think we're going to be just waiting for a few more people to sign on, and then we're going to start. This is Joe Eron. I'm from the University of North Carolina. I think everybody knows Chip Schooley, who's you can see from UC San Diego, where he's driven by something, science, I think.
UC Science Dribble, sorry.
Science driven. And Dr. Ralph Baric, who also happens to be at UNC in Chapel Hill. And literally it's probably the, if not the most, one of the most knowledgeable coronavirus experts in the world, basically. And we're super fortunate to have him here. So Chip, maybe I'll let you take it from here.
Thanks, Joe. I want to first compliment you on the way you made up your bed. Looks great.
Thank you.
And to welcome everybody to our.
Is that better?
No, I actually like it way before. No, I want to welcome everybody in all seriousness to what I think will be a really interesting session. This has been something that we've been wanting to do for some time. Joe and I participated in a session at Croy with Ralph and several other coronavirus experts about what was at that time emerging onto the world scene in early March. And the interactions that we had at that panel were among the most illuminating I've had since the epidemic began. And we've been hoping to have a chance to share some of that experience with those of you who are part of our IAS-USA family and others around the country. So welcome everyone to the colloquium. We'd like to encourage you to engage with us with the questions and answer function that is open to you. And we want to just have an informal discussion about some of the challenges and progress that has been made over the course of the last 8 months. And in Dr. Baric's case, over a career working on these coronaviruses in multiple iterations in multiple species and from multiple continents. So Ralph, welcome. And we appreciate your spending time with us this afternoon.
Chip, it's a pleasure to be here. Joe, it's good to see you again.
Yeah.
Thank you for the invitation to participate, and I'm looking forward to having a pleasant evening with the audience.
So Ralph, maybe you could start, but we'll toss a relative softball to you. What are the features of SARS-CoV-2 that make it so successful as a pathogen? I mean, why are we in this state based on the virus itself?
So the best way to answer that is to contrast it with the SARS-1 virus that emerged in 2003. SARS-1 could transmit fairly efficiently, but it never would transmit until you were very, very, very sick. And so consequently, classic public health measures like quarantine and contact tracing could identify infected patients, identify contacts, get them isolated, and burn the epidemic out. Now, the other drivers for the SARS 2003 epidemic was the animal reservoir of civets and hospitals as acting as major sites for amplification of the disease. So let's compare that now with SARS-2 of 2019. Well, SARS-2 can transmit from patient to patient before clinical disease emerges. Mm-hmm. So it's much more flu-like in its characteristics. And so many, I think on average, I don't exactly know what the RO is, it's bounced between 3 and 5 depending on the study. But in general, most patients will transmit to a couple of other individuals with rare super spreaders infecting 15, 20 people in a very short period of time. And so in the absence of clinical disease, you don't know who's infected. You don't know who they've transmitted to. And you have these silent transmission networks that are spreading out through a nation. In the case of the United States, in the middle of the flu season. And so it was hidden. It was there, the signature was there, but it was very, very hard to see. And so it really grabbed hold of us when it finally managed to get into the country. So that's one factor, transmission prior to the development of clinical disease. Another thing about SARS-2 is it seems to grow much more efficiently in nasal airway epithelium and the upper airway epithelium. We're not sure about things like the soft palate and the hard palate. Soft palate, part of the oral pharynx, which is really important for influenza virus transmission. We don't really know what's happening in that region of the respiratory tract. But in any event, it's replicating in regions where there's very, very efficient transmission that can occur from one individual to the next. In this case, we don't know what the animal reservoir is. And in the hospital setting, again, those silent transmission networks allowed it to get established before we really got barrier nursing up and running and in place. And so things caught us off guard. I would also probably argue that in the United States, we were heavily seeded by a new variant called D614G in February. And this virus, as far as all the data suggests, is it's much more easily transmitted from one person to the next. And so this is what's happening in our country today.
And that mutation, that's in the spike protein, is that right? Is that change kind of the binding properties of the virus, or how do you think that mutation affects things, and does it affect things like vaccines? You know, people are worried about vaccines and whether you know, the virus will kind of escape from vaccine based immunity.
Yeah, just to put it in context for the listeners, what came out of China had a particular amino acid sequence in the spike glycoprotein. And that's what arrived in Seattle and on the West Coast. In Europe, a variant emerged that had a glycine residue in the spike glycoprotein at position 614. Who the heck knows what that does, right? Well, it turns out what that does is that, and we have a paper in this up on bioarchives, which is under review for publication. It does several things. One thing that it does is it allows the virus to get into cells quicker. The infectivity of the virus is higher. A bunch of structural biologists have looked at that mutation. I like to put it in the context of a movie. So I don't know if everyone remembers the movie Alien.
Yep.
You know, the egg pods in Aliens with the flaps that would open up and the alien would come out and grab the person. Well, that mutation basically opens up the receptor binding domain, at least one receptor binding domain, so it's in the open position. And that allows it to grab onto the ACE2 receptor really efficiently. That's the hypothesis. And so it can infect really easily. It's much more likely to grab onto the ACE2 receptor to mediate docking and entry into the cell. And so in the paper that we have done, we've shown that the virus actually replicates more efficiently in nasal airway epithelium in your nose, and cells from the nose, cells from the human bronchial airway epithelial, and even in what are called head-to-head competition studies, where you infect a single culture of cells at 10-to-1 ratios, 10 ancestral strain versus 1 contemporary strain. The contemporary strain will outcompete the ancestral strains and outgrow them within 2 or 3 passages. So they're much more fit. They're at a much more competitive advantage. And in hamster studies, in collaboration with Yoshikaoka, we've shown that the new contemporary strain can transmit to hamsters much more efficiently within the first 2 days. And this is by airborne transmission. So you have 2 hamsters in different cages that are separated by space. And the only way one can infect you infect one, and the only way this one over here can get infected is if there's airborne transmission. And the new variant will infect 5 out of 8 hamsters within 2 days. The old variant, 0 out of 8 in 2 days. By 4 days, they've infected all the hamsters both equally well. But that early transmission occurs with the new variant. So it grabs the receptor better. It enters cells, it's more infectious, and it's more transmissible. And that's why the studies of the evolution of this new genotype have argued that there's about a 30 to 40% increase in the RO, or the transmission rate, from one person to the next because of this mutation. Now, it doesn't seem to affect pathogenesis, so it doesn't seem like it's more virulent, which is good news. And the other thing is that it doesn't seem to affect its ability to be neutralized by antisera. So the current vaccines, the current people who've been infected with the ancestral strain, the first strain that came out of China, they can neutralize this virus very, very well. And in fact, it's probably a little more sensitive.
Right, because it's open.
It's in an open configuration, and there's important neutralizing sites on that face that grab onto the ACE2 receptor. And so it's good news for vaccines. It's bad news because it's much more transmissible potentially. And but then again, you know, we have 7 billion naive hosts on the planet, and so the virus is just, you know, looking to evolve strains that can spread easily and transmit easily. That will probably change if 40 to 50% of us have antibodies against the virus. It's going to cause a different selection pressure.
So what would you be looking for as a molecular epidemiologist about directions the virus might go? I mean, we've talked about its mutation rate being substantially slower than flu and HIV. What other things can the virus do to fix itself up, to help itself around the human population? It's thought this out pretty well so far.
I think, well, you know, there's 40 million of us that have been infected you know, that we know of.
Right, exactly.
It's probably 2 to 3 times that at a minimum of what we don't know. So we're probably somewhere between 100 and 200 million infections on the planet. So the virus has colonized us, and it's not going to stop until either we vaccinate the population or it burns through the population. Those are the 2 outcomes that we're facing. And am I expecting? I'm not really expecting a change in virulence, but this is uncharted territory. So for example, the last pandemic respiratory virus that swept through the population was in 2009 with H1N1. And that was in the context of some pre-existing immunity that could provide some protection. So this is completely unprecedented. So we don't know what's going to happen in terms of how the virus will evolve and colonize the human host. And so we can't really predict it. The other thing that we can't predict is that, you know, this virus presumably came out of Southeast Asia, southern China. It's coming in contact with many, many different mammals that it's never seen. And so it could colonize bats, it could colonize other common mammals, mammalian species, to set up new reservoirs in North America or South America or Europe that are completely unanticipated. And in that context, especially in bats, they can come in, the virus will come into contact with many other bat coronaviruses that set up scenarios of recombination. And recombination is basically a function where a cell gets infected with 2 closely related coronaviruses and they admix their genetic material. And coronaviruses do this really well. And so all sorts of potential exists for the emergence of new variants because of colonization of other species outside of the human host. So it's unprecedented, undiscovered territory. We don't know.
We've certainly seen the evolution of our understanding, at least, of the host range go from bats to humans, now to hamsters, and people are, it's as you said, dogs and cats can occasionally have had virus isolated. Do you see the virus looking for additional species to evolve into over time that could, in addition to bats, serve as like camels do in the Middle East for MERS?
I think we've already seen pretty extensive infections in mink farms, for example. So that's one potential reservoir. Bats are kind of the most logical one. They're certainly the one that's a major concern because of all the other pre-existing bat coronaviruses. You know, each bat species has on average somewhere between 7 and 10 unique bats coronaviruses. So this massive reservoir for genetic exchange and evolution in that environment. Otherwise, there's a large number of mammalian ACE2 receptors that can be used for docking and entry. And so it only takes a single point mutation or two to adapt efficiently to that receptor. And then there are some other host factors that are important for infection and maintenance of the virus in the population, in the animal population. Most likely, the most important being density and the ability to set up a chronic infection. And if those kind of events occur, then we'll have a reservoir.
Yeah, density of bats. I never really thought about that, but that makes a ton of sense. The density of bats. Do you have a sense, Ralph, of why, for example, younger people seem to be less symptomatic? There are all these different studies looking at the proportion of people that become infected and become symptomatic, and it seems like it's really, inversely or directly proportional to age, really. That if you look at nursing home populations, it's 60 or 70%. If you look at, I think at UNC, obviously, with probably thousands of students that have been infected, and I think maybe we've had one or two hospitalizations total of students. What's different? Is it the immune system? What do you think?
Every coronavirus that we've worked with has a really strong age-related disease gradient. In other words, the generally mild. Every human coronavirus that we've worked with, has been generally mild in younger individuals, although the very, very young can be highly susceptible. But in general, the younger individuals have mild disease. And the more elderly you are, the more likely you're going to have severe disease. That's the case with SARS, SARS-2, MERS, even the contemporary human coronaviruses like OC43 and 229E and NL63, in general cause mild common cold infections in young children and adults, except for in seniors. And there have been outbreaks of OC43 in retirement communities with 10% mortality rates, which are equivalent to HKU1, which is another contemporary human coronavirus, was actually identified and misclassified as a SARS case. It happened in an elderly gentleman who died from the infection. So in general, coronaviruses have this strong age-related disease gradient. So the question is why? Well, part of it's probably immunosuppression of the host, of the elderly host. Part of it's related to ACE2 expression. ACE2 expression has a tendency to go lower as a function of age. It's also on a sex-linked chromosome, so males have less ACE2 than females. And too much ACE2 may actually be protective. And so that's one hypothesis. There's the immune suppression hypothesis. There's the ACE2 receptor expression hypothesis. And so those are both factors that could contribute to more serious disease as a function of age.
One of the points you made in the past is that in addition to the age-related pathogenesis, these viruses don't induce immune responses that last very long. And that instead of having to use viral evolution to continue in the human population, the same virus can come back every 3 or 4 years as the immune density declines. Are there strategies to get around that as we're trying to think about vaccines that are going to do a better job of inducing immunity than natural infection, which doesn't seem to be very protective over the long term?
So when you think about modeling where this virus will go, one model that you can use is what do we see? What's the epidemiology of the contemporary human coronaviruses, the ones that we live with every day? Well, those are mostly childhood infections, and they can be severe and cause lower respiratory tract infections in very young children. And they induce a protective immune response, but it does wane over time, but it still provides some level of protection. And so typically the coronaviruses were defined as common cold viruses. They replicate in the nose and upper airways, mild symptoms in most people. Your immune system lasts for about 2 to 3 years, and then you get another infection that is again localized primarily in the upper respiratory tract. And that occurs until you begin to become elderly and immunosuppressed, where your immune system begins to fall off. And then that immunity wanes sufficiently to cause a lower respiratory tract infection that can cause serious disease. So the prediction with SARS-2 would be after the virus runs its course through the population, the virus would maintain itself primarily by infecting newborns, young children, in the first few years of life, and then there would be reinfections that would occur throughout your life, but in most cases, it would be mild common cold-like symptoms. And so that would be the prediction for where SARS-2 would be going. It may just take 5 years to get there. And with a mortality rate maybe around that, nobody really knows what it is. Let's say it's 1%. That can be a lot of mortality on a global scale.
Ralph, there are a couple of questions in the chat.
Sure.
One of them is, why is the rate of transmission in Africa lower compared to the rest of the world? Is that a true difference, or is that a testing phenomena? What do you think? Is there some genetic difference that the virus is failing to exploit, or?
There could be a lot of different factors. One thing that we need to look at is whether, what's the role of the microbiome and chronic inflammation in pathogenesis. So if you have a population that has low level, some percentage of parasitic infections or other chronic infections, how does that influence the disease course? If it is somewhat attenuating, then that could explain the data. It could also be sampling, in that the epidemiology and the testing is not as thorough as we might like in Africa. There's certainly some places that have done a good job of this, but I don't know if you could, extrapolating that to the entire population of Africa based on 6 to 10 sentinel sites might be overinterpreting the data. So I think we still need to look at that in more detail, but it also could be genetic. There's been at least 2 papers published about the susceptibility loci on chromosome 3. That has a set of about 5 genes on it that might be playing a role in pathogenesis. We don't know for sure exactly what's happening and what the role of those genes might be, or how they could be contributing disease, but that's, if there's if the distribution of that susceptibility allele in terms of resistance to infection is higher in Africa than it is in other populations, then that would also explain the data. There's a ton of things that we don't know.
Could be timing too, like when the virus hit Africa, they were kind of moving from spring to summer. The peak, they certainly had a peak when we did, and then they seem to be coming out of it now while we're going back into it. Do you think, does seasonality really matter, or is it just proximity?
I think seasonality, there's seasonality definitely matters, and it matters in a couple of ways. Probably the most important is the potential infectious dose that you might receive might be higher in cooler temperatures and in closer proximity.
Sure.
So in animal models of coronavirus infection, there's a clear infectious dose range that it might require 1,000 to 10,000 virus particles to establish a robust pathogenic infection. And so in summer months, with what you would hope would be populations that are more distributed, more separated from each other, that the infectious dose would actually be lower. And so the disease severity would be proportionally lower. And so that's one of the main concerns as we move into the winter months. Not only what might we see increased transmissibility, but we might see increased viral load that's establishing the infection, in which case the progression may be much quicker. And if the virus doesn't need too much of a head start really get ahead of the immune system and begin to shut it down in that case, then so again, it's undiscovered territory for us, because in reality, our pandemic was really recognized in April as a real problem. And so that would be the time period where the temperatures are increasing, and perhaps the infectious load, the dose that's going from one person to the next, may be decreasing.
The aerosol scientists also talk about indoor heating lowers the humidity, and that dries the aerosol particles more rapidly and allows them to stay airborne for a longer period of time. So you're absolutely right. We could end up with a kind of a real acceleration that even outstrips what the IHME has been predicting.
And the number of asymptomatic infections may change.
Yep, absolutely.
And so we don't know what, we don't know what's going to happen in the winter months. This is going to be the first time we experience it. And in fact, the same with the rest of the world for the most part.
So as with Game of Thrones, winter is coming.
Winter's coming.
Thanks, Chip.
Not a good analogy. Sorry. So one of the other questions in the chat box here has to do with therapeutics. Joe and I are therapeuticists. We would love to see vaccines succeed. What prospects do you see for an analogy to HIV, in which the vaccine promise is still there, but the therapeutics are what change the disease?
Can you ask that question again? I didn't quite follow
In 1984, a couple of very famous people said we'll have an AIDS vaccine by 1986. And we spent a fair bit of time and money on an AIDS vaccine. We don't have one. The face of AIDS changed because of therapeutics.
That's true.
Is it feasible that longer acting, orally bioavailable, or other drugs could be used to take the lid off of the disease in people at substantial risk for the disease, or could be used to interrupt outbreaks, even if we're still playing with vaccines 10 years from now?
Yeah, and oral therapy is the game changer here. A direct-acting antiviral oral therapy is the game changer. And that's because all the animal models agree. And the quicker you get the therapy into the animal, before peak titers hit, or by the time peak titers hit, the more likely you can change the course of disease and end up with a survival situation for the patient. And of course, the problem with remdesivir is the IV drug use, and coupled with, I would guess, a clinical decision about bed space, right? You can't be too aggressive by admitting every patient when you know a good fraction of those patients are going to have mild disease and putting them on therapy. So you have to triage to determine who's going to go in the hospital. And every single day of triage with a direct acting antiviral makes it less and less effective. Yeah. And so an oral therapy that you can take home as soon as you're diagnosed to go on that therapy is a game changer for the entire field, and for the community and for the patients. And it will also probably knock transmission way down, which would be good. So just that's the real game changer. Then the second game changer is, how do we deal with this pathogenic inflammatory response that occurs in the second phase of disease? So I always view it as a biphasic disease. First, associated with acute virus replication, destruction of AT2 cells, or alveolar type 2 cells, which can lead to all kinds of problems with oxygen, lack of oxygen, and flooding of the alveoli, which is generally never a good thing to have happen. And then as the virus is cleared by the immune response, you have this immunopathologic response, which also causes damage. And so how do we control that? And there's a lot of anti-inflammatories being tried. Dexamethasone looks like it works well late in infection. Of course, if you give it too early, then you immunosuppress and the virus wins. So
we had several questions, Ralph, about that. about using dexamethasone in outpatients. These were questions submitted. And I think in the recovery trial, as you just pointed out, people with moderate disease who got dexamethasone actually had greater mortality. It didn't quite reach significance when they adjusted for it, but clearly there was greater mortality. And it seems like in the beginning, there's this kind of race between, viral pathogenesis and development of antibody, or an immune response. I don't know if it's antibody that's important, but the recent data from Regeneron suggesting that people that were symptomatic and had antibody were much less likely to go on to have illness, where those who were at similar degrees of symptoms but had not yet generated antibody. And a therapy, as you mentioned, that would kind of slow the pace of the virus so the immune system can kind of catch up, would, like you said, be a game changer, I think.
Right. I think anti-inflammatories will work well used sequentially with a direct-acting antiviral. So if you can put them on a direct-acting, like an oral direct-acting antiviral, or even a therapeutic antibody early enough, and then come back through with an anti-inflammatory, that combination where you're controlling virus replication and simultaneously trying to damp down a little bit on the immune response may be the right cocktail. The downside of that may be that those patients don't mount very good immune responses from infection, and then they could be susceptible again. So that would be that's sort of like, we don't know the consequences. But then again, you treat the disease you're facing, because you, at least if patients don't survive, you don't ever have to
deal with the kind of second and third problems. That there's a lot of discussion about these kind of longer term side effects. People are calling them long haulers. People who seem to recover from the acute infection and even from the more life-threatening immune response, but then have these persistent symptoms. I assume that these people have cleared their virus, or do we even know that?
My prediction would be that they've cleared the virus and it's set up some sort of chronic inflammatory disease process that could progress to pulmonary fibrosis or some other chronic lung manifestation. There's a lot of damage occurring in the alveoli, and A22 cells, and the other cell that other stem cells probably in the lung are being infected, which affect repair processes. And so there's probably all kinds of chronic lung manifestations that are either going to take a long time to recover, or perhaps in some cases, the virus may well have killed you. It just takes another 4 years for that to occur. Sort of a hit and run type disease.
Yeah. Sorry, go ahead, Rob. Do we understand some of the, are there animal analogs to some of the vasculopathy that people see in lungs of people with severe disease in addition to the airway disease?
Not yet, I don't think.
Okay.
And do you think that kind of intrapulmonary and sometimes extrapulmonary coagulopathy, that is that a direct infection of endothelial cells that also express ACE2, or is this another immunologic phenomena?
So as far as we can tell, using both primary lung endothelial cells and animal models, the virus doesn't seem to infect endothelial cells that well. So it's probably some sort of secondary mechanism. In terms of the coagulopathies, we've seen the coagulopathy with SARS-1 infections in mice. We have not, we're very interested, obviously, in doing those similar types of experiments in our mouse models that we've developed, and those experiments are progressing, but it's not like I can tell you that we have definitive proof that there's a coagulopathy that's occurring in the context of infection. But I think there is, personally, in the mouse model. It makes sense. I think SARS-1 and SARS-2 have very similar pathogenic models, and In mouse models, for example, where we map susceptibility loci that regulate SARS coronavirus pathogenesis, a lot of those same susceptibility loci seem to be important for regulating SARS-2 pathogenesis as well. So I believe there's a coagulopathy in the mouse models. I just haven't published a paper proving it yet.
So somebody has a question about the paradoxical, paradoxical observation that of more ACE2 receptors, less ACE2 receptors, and disease severity. If you have more receptors for the virus, why would the disease not be more severe in that setting and progression more rapid?
Yeah, so there's a fellow named Penninger who did some really interesting experiments between 2003 and 2010, where he showed that high levels of ACE2 expression are actually protective for not only SARS coronavirus pathogenesis, but also flu pathogenesis. So ACE2 is an important, is not only important for regulating homeostasis, but it's also some of the cleavage products can signal down through various pathways to turn on inflammatory processes. And so high levels of ACE are actually protective to some extent in disease models. And if you knock out ACE2, for example, in a mouse and infect it with flu, the animal does very, very poorly. So ACE2 is not just a receptor for entry, it's an important signaling molecule that can regulate inflammatory processes and other processes that can lead to disease. So the feeling is high levels of ACE2 set up a signaling pathways that are more protective and less inflammatory. That's the theory anyway.
It makes more sense than trying to make a direct tie into the receptor density, and that is the only role for that molecule. Certainly does. Yeah. Interesting. The people here are asking, obviously, about the question about laboratory creation of the virus. Which laboratory do you think made this virus?
Right now, there's no evidence that any laboratory made the virus. The virus's closest relative is a bat virus called RATG13. It's 96% identical to it. If I remember correctly, HIV was supposed to be a laboratory-acquired generated virus. H5N1 was a laboratory-generated virus, and SARS was a laboratory-generated virus, and now SARS-2 is a laboratory-generated virus. And to begin with, pathogenesis is an incredibly complex process, and it's almost like a symphony. And all the instruments have to play exactly right to cause disease. And we're not good enough to do it by straight engineering type processes. And the second thing is, we've had 8 pandemic viruses in the 21st century, all of which came out of animal reservoirs. So is it really surprising that another one came out of an animal reservoir? All the contemporary human coronaviruses can be traced back to strains that existed either in bats or in rodents and cattle. And so zoonotic viruses spill over into human populations. They spill over into other animal populations. There are 7 billion of us on the planet. We are in close proximity and high density in regions where many of these zoonotic viruses live in Southeast Asia and in Africa and in South America and in the United States. And because we're naive, eventually a virus figures out a way to colonize a new host. That's what they do. And they do it very well. And RNA viruses have a high mutation rate. And so they exist as a swarm of variants. And those swarm of variants can rapidly adapt to a new species.
Ralph, one thing about RNA viruses is, we talked about an oral antiviral being a game changer. I mean, there's a lot of RNA viruses we'd like to have a really good therapy for. And I assume people have been trying to develop therapies, obviously for influenza for sure, but for rhinovirus, you know, and it seems like the polymerase of the coronavirus is not so easy to kind of attack with small molecules. Is that a fair statement, or is that just people haven't looked hard enough yet?
Well, so in the case of coronaviruses, as far as I know, there were only a couple of groups in the world that were actually looking for antivirals against coronaviruses, and certainly Mark Dennison's group at Vanderbilt and our group were probably the major groups globally. You know, many companies started to work with developing antivirals against SARS, but they stopped within about 6 months of starting those efforts, and all that work went onto a shelf and they forgot about it. And so the view that emerging coronavirus could cause a pandemic with high mortality was just not viewed as a credible threat. And despite the fact that SARS coronavirus had emerged, infected 8,000 people and killed 800, and that MERS coronavirus has a 35% mortality rate. And we know that RNA viruses can mutate, and it doesn't take very many mutations to go from I'm trying to figure you out to I own you.
Right.
And because they make so many mutations, when they replicate, they sample a lot of diversity. And so there weren't very many groups working on a broad spectrum coronavirus antiviral. I would say the rhinovirus, and another example, take rhinoviruses. There was a lot of effort early on to develop a compound that would work against the common cold. But the major problem is that when common cold viruses infect you, by the time you know you're sick, most of the damage has occurred, so the antiviral doesn't work. And so that's what happens with many respiratory viruses, even influenza viruses.
Influenza, sure, yeah.
After day 2 or 3, it is a crapshoot on whether or not that drug's doing any good or not in the patient.
So here's a virus actually where we know that there's substantial replication in people before they become symptomatic. So I guess that would be an opportunity. Are you actually optimistic about an oral antiviral, or
we've published on one that is currently being developed by Ridgeback Biosciences in collaboration with Merck, and it's in phase 2 trials and in several locations around the United States. It works great in animals and in primates, but I don't know, I don't know where the things stand right now in humans. We don't know.
You're making a great case, though, for a more general need to have a longer attention span than we do as a biomedical research community.
No kidding. I mean, 8 epidemic or pandemic viruses in 20 years, that's every 3 years.
Right.
So if we project forward over the next 80 years, what is that? 25 more pandemic viruses? Come on, we need to wake up. We need to do two things absolutely critical. We need to strengthen our public health infrastructure and really provide support for agencies like the CDC in terms to gear up their surveillance to make it 21st century like. In other words, take advantage of the internet, take advantage of social contact networks, take advantage of pathogen genome sequencing for contact tracing at a granular level that makes a difference in terms of an expanding pandemic. And then the other thing we need to do is we need to develop drugs for broadly active drugs against these sort of pandemic virus families like alpha viruses, flaviviruses, coronaviruses, influenza, the paramyxoviruses, filoviruses that we may have a good handle on, but we still need a little bit more, and the hantaviruses. And these are, I apologize to any virus family group members out there studying some viruses that I forgot, and now I'm going to get these terrible emails over the next here's it, read this paper. It's like, I just forgot it. But certainly all of the RNA viruses have the potential to be problematic, and we should recognize that, and we should get broad spectrum drugs on a shelf. We can do this. This is not an unsolvable problem.
Yeah.
This is just, let's put some resources here so that we don't ever have to face this again. So public health practice, public health coupled with drug development, absolutely essential.
Well, there's a question from Doug Richmond about TMPRSS2. Do you have any comments about its role in pathogenesis of the disease?
Yeah, so there are two major determinants for for regulating how coronavirus gets in a cell. The first is the receptor molecules, and the second are the proteases that have to cleave that spike to mediate entry processes. And TMPRSS2 is a major player. Now, the funny part, of course, is that not all coronaviruses are absolutely critical, require TMPRSS2 to mediate docking and entry. And so we can knock out TMPRSS2 in a mouse and still infect it just fine with SARS-1. Didn't say SARS-2, SARS-1. And so there is actually a slew of proteases that could potentially be used by the virus to mediate entry into the cells. And different sarbo-coronaviruses, SARS-like coronaviruses, have different cleavage sites that allow them to have more than one way into the cell. So for example, if you just target a single protease as a drug against a coronavirus, you may find that it attenuates replication in one cell type, but allows it to replicate in many other cell types. Mm. So protease inhibitors are a little bit of it's not a one gene, one drug problem. It's a complex problem, and you need to think about it really carefully.
Ralph, we have a ton of questions about vaccines. Do you want to comment about whether you think that we'll have a successful vaccine? We just, in the New England Journal, we just saw that the mRNA vaccine protected primates in a challenge model. But, in reading that, I can't tell whether that was a softball, it was an easy win for a vaccine, or should we be heartened by the fact that primates were protected? Or
so the primate models with SARS-2 are they're not rigorous. They develop a mild disease. You have to put 10 to the 6th in to get any kind of disease signature. Most of the readouts are in RT-PCR genome equivalents, which aren't that high, which means the virus titer is probably 2 to 3 logs lower than what they're showing in the paper. And so there's no question that vaccines work pretty well, but is that a good model for what's happening in a human? I'm not 100% sure of at this time that it's a great model. Part of that may be because many of the viruses that are being used in those models have undergone 4 to 5 or 6 passages in cell culture, and they have deletions in the furin cleavage sites and point mutations, or deletions in the NTD domain and elsewhere that could affect pathogenesis. And so they're trying to work that out now with a more uniform challenge stock that may be more pathogenic. So I'm not sure whether it's the model or whether it's the viruses being tissue cultured, adapted, that went into the model that are causing the problem. The hamster model is pretty robust. The animals lose 15% body weight. They get pretty sick. The pathology looks good in the animal. There have been several vaccines, I think, that have worked in that model. The new mouse models, the ACE2 transgenic mouse models, all suffer from a mild respiratory tract infection followed by invasion of the central nervous system and death by encephalitis. Whether they're willing to admit it or not, depending on the model. And then the new mouse adapted model that we made is a lethal model with a more acute lung injury, ARDS phenotype, with lots of surfactant C, and which would result in a surfactant deficiency that could. So it's more like a severe respiratory tract infection. The Moderna vaccine was tested in primates. It's certainly protected there. I think the better data is the New England Journal data that showed that the vaccine induced really high neutralizing titers, as well as various T cells, high neutralizing titers in human, especially aged individuals.
Right.
Where they look at and they compared the neutralization titers to individuals who were infected with the virus naturally. And in that case, the vaccine produced responses that were in the top quartile. In other words, the top 25% of the responses. So that's really encouraging because, you know, the aged population it's a difficult population to vaccinate well. And the fact that the mRNA vaccine worked well in that is a good sign.
Mm-hmm.
Now, does that mean it's going to protect? I don't know.
Sure.
But it's a good sign. The mRNA vaccines that we published with the NIH, with MERS, which is a lethal disease model with 30% body weight loss and a full ARDS phenotype, completely protect those animals, young and old animals. So it's a pretty rigorous vaccine, I think. Again, you never know until you see the human data.
But there are animal coronavirus vaccines, correct? That work. I mean, there's a model that someone asked about. You know, we haven't developed an HIV vaccine. Why should we be, you know, thinking we're going to develop a SARS-CoV-2 vaccine? I mean, there is some.
Well, let me start out by saying that HIV is a very different beast.
Right.
And the mutation rate for HIV is 10 to the minus third. So 1 out of every 1,000 bases is a mistake. 1 out of every about 1 million bases is a mistake with SARS-2. So the evolution rate is different. HIV is attacking critical immune effector cells that regulate the whole immune response. SARS-2 is not. It's replicating in the lungs. So there's no reason why we can't create a vaccine that works against SARS-2. And HIV is the vaccinologist's worst nightmare, quite frankly. It's just a terrible disease. The virus arsenal that attacks the immune response is unprecedented, and its capacity to evolve and escape is unprecedented. And so that makes it very, very difficult to handle. That's not the scenario with coronaviruses. Now, have there been good coronavirus vaccines? The truth is, the answer to that is mostly no.
I tried.
But you know, there has been this feline infectious peritonitis virus experience, has there not?
That's correct. That's
Yeah.
correct. And so once they got the vaccine, so there's 2 viruses, a feline enteric coronavirus and feline infectious peritonitis virus. They're basically the same thing, but one is associated with an antibody enhancement phenomena where the infection progresses to a situation where the antibody allows the virus to infect macrophages and other immune cells that it normally can infect, and that causes everything to go bad. And if you vaccinate correctly, you can prevent that disease from happening. And so that's one example of where we've had some success. We've also had success in pigs and swine, and also with infectious bronchitis virus, but it's limited success. So the swine coronaviruses typically sweep through and cause 90% mortality in young newborn piglets. And you can't vaccinate the newborn piglets. You have to have lactogenic immunity from the sow. And so the only thing that really elicits a good lactogenic immune response, so that there's high levels of neutralizing IgA antibodies in the mother's milk, is through a live attenuated vaccine of the sow. And so that's provided some protection as well. But so there are ways to make vaccines that work, but not at a so I like this is like, so it's not really at a marketable scale where the industry is happy with the outcome.
Yeah. Okay. Good.
And there's such a pressure to develop a vaccine and release a vaccine before we know for sure how well it works. And as someone who's trying to help with these vaccine trials, once there's a vaccine that's available, the ability to test new vaccines or better vaccines becomes more and more challenging. So It really is a conundrum about, when is the right time to say, this vaccine is good enough. Do you have a sense, Ralph or Chip? You've thought about vaccines forever. I mean,
yeah, I can. I don't know. Chip, do you want to go first, or I've been talking a lot? I don't mind waiting a second.
No, they listen to me all the time. We rarely get to hear what you think, so please.
Okay. So I think Operation Warp Speed has 7 or 8 vaccines that are moving forward, and one of the biggest dangers, I think, is early release of data that suggests that the vaccine is effective based on a few cases, and I'll say why. Most of those cases will be mild infections, so we won't really know whether the vaccine is working in the context of a severe infection. Most of those cases will not be in the elderly, since that fraction of the vaccine population is limited, and there's limited representation of minorities and other ethnic groups. And so that's going to take longer for those data to accumulate to get a really good idea of vaccine efficacy across the population. So the second thing I think that's really important is that even if you know pretty well that a vaccine is working by, let's say, December 1st, the next question is, well, when is the vaccine going to be, when are the doses going to be available? So if the doses aren't going to be available until February, then let's not release the information in December. And because that's immediately going to cause a collapse of all the other studies.
right.
I have volunteered for a vaccine, for example, and so one of the questions I asked was, if we find out a vaccine is working, will I be told whether I was a placebo and can I get the vaccine, or is that information going to be withheld from me? And my understanding is that it's not, which means
He's looking at me.
I'm looking at Joe, and he told me it's not. No, but that means that the placebo controls may immediately flock to the new vaccine, which is going to underpower all the other vaccine studies. And so let's not do that, and unless we know A, the vaccine works really well, B, to give the other vaccines time to accumulate data so that we can have some idea of efficacy. I'm not going to say safety or durability, because we're not going to know that. We're going to know efficacy, short-term efficacy, and then when are those doses going to be available for distributing to the population in sufficient numbers to make a difference? And if it's February 1st, then let's hold off, because we have different vaccine formulations. We got the mRNA-based vaccines, which require a critical cold chain, which means that they're not going to be too effective in Africa or other places in the world that don't have cold chain custody, can't maintain cold chain. We've got some vaccines that are single dose only, and recombinant protein vaccines, which would be very stable, that could be used across the world. So we need time to accumulate that data. And so I think those are factors that need to be taken into account so that we have some idea of efficacy. And I just mentioned that we won't know about safety until we've had a time for vaccine immune responses to potentially wane and for reinfection to occur. And if reinfection can occur 6 months or a year later, that's where we may see a safety signal.
Yeah, there are quite a few questions in what we were sent pre-meeting about this issue of reinfection. I mean, Chip could comment, because I think he's published several of them in his journal, but it looks like there probably are well-documented reinfections, not 100 of them, but certainly multiple ones that look very real. Can you comment on that? And is that 5 cases out of 40 million, or is that just the tip of an iceberg of multiple reinfections and
so both China and Korea reported hundreds of reinfections. Now, they weren't able to validate it because the virus was the ancestral strain that was virtually identical. And so we've now had at least 2 cases where individuals were infected with the ancestral strain and then reinfected with the new global genotype variant. And so that data argues that reinfections can occur. And then there was a reinfection in the Netherlands of an elderly woman who was immunosuppressed who died from the secondary infection. I think the other 2 cases were milder after the first infection, if I remember correctly. So the prediction again would be with any respiratory virus to have sterilizing immunity in the nose and the upper airways is almost impossible for long-term duration. And so it wanes, from my understanding, is it wanes at different rates in the lower respiratory tract versus the higher respiratory tract. And the nose has its own lymphoid tissue that is important in making an immune response in the nose, and it wanes quicker than it does in the lower respiratory tract. And so, you know, you can get reinfected and you have a mild upper respiratory tract infection. The host is okay with it because it, you know, you get to stay home and watch TV. You know, you don't have to go to work. You're not that sick. And the virus is happy because it can replicate and transmit. So it's a sort of a commensal. Is that the right word?
Yeah.
It's a happy relationship between them.
Sure. It's a,
yeah. So that's what I would anticipate that would happen. I don't know what that cycle is, and that's why that, you know, the longer term durability of these vaccines are going to give us a lot of information about, you know, what is the protective immune response, at what level does that fail, and if we have that information, then we know how to do, you know, we know what sort of timeline might be necessary for boosts to occur in the population.
Wow, you're going to lose your voice. We're really working you out. Chip, any additional questions? We're actually a little bit over an hour.
No, I think we really should give him a rest and let him stop aerosolizing whatever virus he's carrying, and go and join the UNC students for a coronavirus transmission experience. But it's been great.
Do my part, right?
Right, got to do your part. You're part of the vaccine study. You'll help Joe out as well if you go down and get yourself exposed. No, I'm being facetious. It really is has been a great opportunity to learn a lot about the long experience you've had with these viruses, because they're really quite germane to the way we're thinking and what the challenges are. And we appreciate all the work that you and your colleagues have done and are doing to help us dig out of this. I think the thing that, if anybody remembers from this, is the point you made that there are some things that are a surprise, and this shouldn't be one of them, that we're dealing with this now. And we're going to be dealing with this for a long time, and with similar events like this, as long as there are humans on the planet. And we need to start thinking about longer term solutions rather than just dealing with last month's virus, which is the way we've approached things.
That's right. A reactive response is a painful way to respond to a pandemic. You want a proactive response so that you are somewhat prepared and have things in place. And again, I can't reiterate enough the importance of public health preparedness, an appreciation by the public for public health practices, an appreciation for the importance of masks and social distancing and preventing transmission. Because right now, best case scenario, before a vaccine comes out, we are still looking at 200,000 deaths, maybe 250,000. Best case scenario when the vaccine comes out. The only way to stop that is going to be public health practice. Wearing a mask, thinking about how you be safe, not being cavalier about the potential danger of the virus. Because again, for most of us, it's not going to be us that pays the penalty. It's going to be our grandparents, our parents, other elderly people, people who have some underlying disease, comorbidity, perhaps a friend that is going to pay the price for your behavior. So for God's sakes, think about the people around you.
Wow.
We couldn't agree more. This is not research that needs to be done. It's been done already. These interventions work, and it's our responsibility to promote them and to take them seriously. So thanks for that eloquent comment to end a really interesting hour, and we hope to see you again for more of these in the future. But thanks for taking the time from what you've been doing, and much appreciated by all of us.
Well, this was a great audience. I really enjoyed it. It was fun talking with you, and we got to laugh some and talk some science, and hopefully informed people who wanted to sit in and listen to us generate a lot of hot air, right?
Yeah, it was a great audience. So thanks, Ralph. Appreciate it.
Thank you. Thank you for attending.
Bye.
Bye-bye.
That was great, Ralph. Amazing.